Hello guys, welcome back to Biolab. This is Vamsi here at Biolab, and in today's video we'll be discussing growth in plants. So yesterday while discussing about the introduction, I have said you that growth in plants is indeterminate, and I have explained you the meaning of these words indeterminate, and also I have said you that the growth in animals is determinate and plants also show determinate growth, but most of it is indeterminate growth. They grow all throughout their life, so that is the meaning of indeterminate growth. So in today's video we will be seeing the types of growth in plant and what kind of growth is seen in plant, and also we will be seeing about the phases of growth and growth rates. So this is gonna be a little lengthy lecture, but nonetheless let's continue. Yes, the growth in plants is, first of all, it's open. Plants exhibit open growth. What do you mean by open growth? No, see, open growth here, it means that every time, every other time, a new cell is added. New cell is added. So what is open growth? It is the addition of a new cell by the meristems. Who does this activity? Meristems. Yesterday we have discussed. They divide. Meristems divide. Okay, meristems divide. Now this open growth is also of two types. Open growth generally can be classified as two types. One is primary growth and second one is secondary growth. What is primary growth, secondary growth? Very easy. You all can understand primary growth and secondary growth. Primary growth, at the end of the yesterday's lecture I have explained you. I am just explaining it again. It is the elongation of plant along axis. Plant along axis. Plant growing along axis this way. The secondary this primary growth is caused by meristems. The secondary growth, it is the lateral growth. You can say lateral growth. You can also say girth increases. Girth means it is the lateral side. If this is the plant grows this way. The plant grows horizontally on x-axis. If primary growth is on y-axis, this is secondary growth is on x-axis. Okay, this secondary growth is caused by the mature cells, or you can say it is generally exhibited by the cambiums. Secondary growth is generally exhibited by cambiums. So this secondary growth is caused by cambium. It can be cork cambium or interfascicular cambium, or you can say vascular cambium generally. Cork cambium and vascular cambium. Cork cambium CC and vascular cambium CV or VC. Yes, these cambiums are developed in gymnosperms and also as well as in dicots. So the plants which exhibit secondary growth in general are dicots and gymnosperms, because they only develop these cambiums later. After their growth, they develop these cambiums. Okay, that is the thing. Primary growth and secondary growth. Primary growth is exhibited by every plant. Secondary growth is exhibited by dicots and gymnosperms. This is the growth in plants. Now let's see the phases of growth in plants. Okay, phases of growth. This is a little important thing. They can ask you a little question from this. phases of growth. See, these phases of growth, first of all, in order to measure the growth in plants, we have a technique called as parallel ray technique. Parallel ray technique. What is this parallel ray technique now? So now see here. Here I have a germinating seed. This was the initially germinating seed and this was later sometime T you see. Now in this parallel ray technique what I do is I just draw parallel rays now. See, this is A, name it A. This diagram is given clearly in your NCERT as well as in the notes which I have uploaded. If you want the notes, as usual, text on my number, you can get the notes. Yes, our website is launching very soon, guys. There also you can access the notes and all the videos will be also posted on the website too. So this is the parallel ray technique. I am drawing parallel rays and I can find which is more deviated from the part. From here I can see that C and D are more deviated. So hence I can say this part, let me change the color. This part of the seed has shown more growth, See, this was initially like this, and if you say after some time it became widened. So this is the parallel ray technique in order to see or check whether the growth is happening or not. Let's see the phases of growth now. These phases of growth include generally 3 phases which you have already learnt in your lower classes. The first one is meristematic phase, second one is elongation phase, third one is maturation phase. Let's see that. Meristematic. Today the stylus isn't working properly, so that's the problem. Meristematic. Let's see one by one. Meristematic phase. Here the work is of meristems. Meristems, what do? They continuously divide. It is a dividing phase. Remember, it is a dividing phase. Of the cell. During this phase, the cell has some important conclusions, it has a very dense protoplasm. Let me draw the cell first of all, then I will be explaining you from that diagram. So these are the 3 phases: meristematic phase, elongation phase and maturation phase. Now let's go one by one. I have drawn the diagrams for everything. I am just explaining you. Now see, this is the cell during the meristematic phase. This phase is a dividing phase of the cell. Cell divides actively. Cell divides actively. And it has thin cell wall, as you can see. When compared to the original matured plant cell, it is having thin cell wall. It has no vacuoles. You have to remember, no vacuoles. It has many nuclei. We have already seen growth is an expenditure process. Which expenditure process? Energy expenditure process. So for that reason it is having many nuclei. Dense protoplasm, very dense protoplasm. Protoplasm, it is the sum of nucleus and the cytoplasm. So total inside material is called as protoplasm, and these cells are present at the apices of both root and shoot apex. So this is the meristematic cell, how it looks and how it divides. So now let's move on to the second phase, elongation phase. In the elongation phase you can see some changes. There are vacuoles forming. See. So 1 minute, guys. there are vacuoles forming. See here, there are many small vacuoles. Thick cell wall decomposition has started. Thick cell wall is getting decomposed. More cellulose material is there, and also cell is elongated. Cell is elongated or enlarged. Nuclei number is decreased. Nuclei are less when compared to the previous one. Protoplasm density also decreases. Less dense protoplasm, relatively less dense. Less dense protoplasm, relatively it is less dense than the meristematic phase. So as you can see, number of nuclei are decreasing. So from this you can learn that the cell is reaching its maturity. The phase which is in between the juvenile period and mature period is the elongation phase. In our humans, the juvenile period and the adult phase is, the gap is covered by teenage. So like that also, this is a teenage, you can remember like that. Teenage for plants is elongation phase. Thick cell wall decomposition, number of vacuoles are forming, nuclei are decreasing, protoplasm density is also decreasing. Now the maturation phase. This phase is the matured phase of the cell. Matured phase. This is a matured phase. It is having thick cell wall decompositions, a large vacuole, large central vacuole, peripheral nucleus, sorry, peripheral nucleus, and also protoplasm density has attained its minimum phase, minimum dense protoplasm. The protoplasm density is very less when compared to the meristematic phase. The density decreases, thick cell wall decomposition, and this is the maturation phase. And here the cells are converted into permanent cells and they attain maturity, and also they have a specialized functions. So this is the phases of growth. Now let's see the growth rate. What is growth rate? Yes, growth rate. For a minute, please think what is meant by growth rate. You would get, pause the video and think what is growth rate faster. Yes, growth rate. It is the growth per unit time. Growth per unit time is growth rate. Growth per unit time is growth rate. And this increase in growth means increase. So this growth rate can be of two types. One is arithmetic growth rate and another one is geometrical growth rate. Let's see what is this arithmetic growth rate and geometrical growth rate. Let's clear the page. Yeah, complete it. Now growth rate, arithmetic growth rate, geometric growth rate, GGR and AGR. Arithmetic, geometric. In arithmetic growth rate, when one meristematic cell divides into two, one of it is differentiated, means it gets converted into permanent tissue, and other one keeps dividing. See, again after this is divided, this cell is one, this gets differentiated. So two cells get differentiated. Here one cell is differentiated, here two cells is differentiated. So that is why the sequence is 1, 2, 3, 4, 5. It continues like that. Hence this is an arithmetic progression. Therefore the growth rate is called as arithmetic growth rate. the growth rate is called as arithmetic growth rate, and the cells adding are also in the same proportion. The cells which are adding are also in the same proportion. So that is arithmetic growth rate. One cell divides, other cell matures. Now geometric growth. Here no maturity happens. If one cell divides into two, no maturity, and these cells form again 4 cells. See, this forms 2 cells, this forms 2 cells. Here 1 becomes 2, 2 becomes 4, 4 becomes 8, 8 becomes 16, 16 becomes 32, 32 becomes 64, 64 becomes, and so on. That continues, that continues. Hence this is a geometric progression. Therefore the growth rate is defined as geometric growth rate. This is geometrical growth rate or geometric growth rate. Hope you understood the differences between the arithmetic growth rate and geometric growth rate. Now let's try to express these growth rates as an equation in the form of an equation. Let's try to express them. Cleaning. Yeah, arithmetic growth rate can be expressed with an equation LT equal to L0 plus RT. LT equal to L0 plus RT. LT is the length of the plant in time t. L0 is the length of the plant when t equal to 0. Time t is equal to the initial length of the plant, and R is the growth rate, T is time. Now let's see how geometric growth rate can be expressed. Geometric growth rate can be expressed with an equation. Geometric growth rate can be expressed with an equation W1 equal to W0 e to the power RT. Here we are using exponential. So that is why geometric growth is also called as exponential growth. So now, yeah, finished. Let's finish first. This is the final stage. This is the final size. This is the initial size. This is the exponent or base e, and it is a base of natural logarithms, and R is the growth rate, T is the time. Now let's plot these on graph. Yes, I have 2 graphs here. The arithmetic growth rate comes with a straight line, and geometric growth rate is an S-shaped curve, sigmoid curve, which represents the 3 phases of growth. See, this phase is the lag phase. The cells divide slowly. Why they divide slowly? Since there are less number of cells. After reaching some amount of cells, they divide faster. Exponential growth is seen. This is called as log phase, and after some time the growth stops, and that is represented, the growth becomes constant, and that is represented as stationary phase. So same thing you can relate to lactobacillus lab bacteria in the curd. When in the curd preparation, when this lab is added as an inoculant into milk, the initial time this lab bacteria divides. Since there are less number of lab bacteria, they divide rapidly, and cells, they form new cells. After that there are a little amount of cells. Each cell divides, forms this much, and finally they cannot divide much, so they stop their division, and hence curd is prepared. So this graph can be is a sigmoid graph. The geometric growth and arithmetic growth rate graph is a linear curve. Please remember this. So that's it for this video. In the next video we will be talking about differentiation, and thank you for watching my video with patience. This is Vamsi signing off from Biolab. If you like the video, please hit that like button. Also subscribe to our channel Biolab. Thank you guys. Have a great day further.